Synthetic Human Embryos, Pioneering Science and Ethical Quandaries. Introduction. The recent announcement of the creation of synthetic human embryos using stem cells has ignited a passionate debate within the scientific and ethical communities. These synthetic human embryos, often referred to as SHEEFs, synthetic human entities with embryo-like features, are remarkable structures that mimic early human embryos. However, what sets them apart is their unique origin. They are constructed from pluripotent stem cells without the involvement of egg or sperm cells. This groundbreaking achievement holds the promise of providing crucial insights into human development, genetic disorders, and pregnancy loss. Yet, it simultaneously raises complex ethical and legal questions surrounding their creation, use, and regulation. What are synthetic human embryos and how are they made? Synthetic human embryos, SHEEs, are not the result of the natural fusion of egg and sperm cells. Instead, they are crafted from pluripotent stem cells, which have the remarkable ability to differentiate into almost any cell type in the human body. These stem cells can be derived from embryos or reprogrammed from adult cells, such as skin or blood cells. Creating SHEEs involves manipulating the culture conditions and signaling mechanisms that guide cell differentiation. Researchers can coax these stem cells to self-organize into three-dimensional structures that mimic various aspects of early embryonic development. These structures can simulate features like a blastocyst-like cavity, placenta-like tissue, and a primitive streak-like structure, marking the onset of gastrulation, the process leading to the formation of the three primary germ layers, ectoderm, mesoderm, and endoderm. The first SHEEs were reportedly created by a collaborative team from the University of Cambridge and the California Institute of Technology. Their work was unveiled at the International Society for Stem Cell Research in June 2023. These synthetic human embryos were cultivated to a stage equivalent to just past 14 days old, which aligns with the legal limit for studying natural human embryos in many countries. Rules governing synthetic human embryo development. The regulations governing the development of synthetic human embryos vary significantly among countries and regions worldwide. Some nations, such as Germany, Italy, Ireland, Poland, and Slovakia, maintain stringent prohibitions or restrictions on all forms of human embryo research. Conversely, others, including Australia, Canada, France, Japan, Singapore, South Korea, Sweden, the UK, and the US, have more permissive regulations allowing specific types of human embryo research under defined conditions and rigorous oversight. However, most existing regulations do not explicitly address synthetic human embryos or other stem cell-based embryo models. In India, for example, there is no dedicated legislation regulating synthetic human embryo research. Nevertheless, there are general guidelines governing stem cell research issued by the Indian Council of Medical Research, ICMR, in 2017, which lay down ethical principles and norms for conducting stem cell research involving humans or animals. International agreements concerning synthetic human embryos. At the international level, there is no binding treaty or convention specifically regulating synthetic human embryo research. Nevertheless, non-binding declarations and recommendations offer some guidance and standards in this field. Notably, 1. The Universal Declaration on the Human Genome and Human Rights, UNESCO, 1997. This declaration emphasizes the prohibition of practices contrary to human dignity, such as reproductive cloning, and highlights that interventions on the human genome should only occur for preventive, diagnostic, or therapeutic purposes with informed consent. 2. The Universal Declaration on Bioethics and Human Rights, UNESCO, 2005. This declaration underscores the full respect of human dignity, human rights, and fundamental freedoms in scientific research involving humans. It prioritizes individual interests and welfare over the sole interest of science or society. 3. Guidelines by the International Society for Stem Cell Research, ISCR, issued in 2021. These guidelines offer detailed recommendations for ethical and responsible stem cell research involving human embryos, stem cells, organoids, and other models. The significance of synthetic human embryos. 1. Study of human development. synthetic human embryos serve as a powerful tool for investigating human development, particularly during early stages that are challenging to observe in natural embryos. Researchers can use them to understand cell differentiation, tissue formation, gene regulation, disease causation and prevention, and the establishment of pregnancy. 2. Alternative for research. Synthetic human embryos provide a valuable alternative or complement to natural human embryos for research. This lessens reliance on donated IVF embryos, which are often scarce, and helps address ethical concerns related to their use or destruction. 3. Applications in regenerative medicine. These embryos open new possibilities for regenerative medicine and biotechnology. They can be employed to generate specific cell types or tissues for transplantation or therapy, such as blood cells, nerve cells, heart cells, and liver cells. Additionally, synthetic human embryos can be used to create models of disease or injury for drug testing and screening. Issues surrounding synthetic human embryo development. 1. Unclear regulations. The creation of synthetic human embryos raises complex ethical challenges concerning their moral status, utilization, and regulation. These questions include whether they possess interests or rights requiring protection, how they should be governed and employed, who should have access to them, and who should oversee their use. 2. Misconceptions and public perception. The development of synthetic human embryos may provoke negative reactions from segments of society who perceive them as unnatural, immoral, or dangerous. Moreover, they may generate unrealistic expectations or false hopes among individuals seeking them for unproven or inappropriate purposes. 3. Cloning and safety concerns. There is significant uncertainty regarding the long-term effects and consequences of manipulating stem cells to create synthetic life forms. Extensive testing and monitoring are essential before applying these technologies to humans or animals. Addressing issues related to synthetic human embryos. 1. Comprehensive discussion. Ethical questions surrounding synthetic human embryos are multifaceted and divergent. Therefore, stakeholders such as researchers, ethicists, policymakers, regulators, clinicians, patients, donors, advocates, media, and the public must engage in extensive dialogue on debate to navigate this complex terrain. 2. Collaborative efforts across regions. Given the global implications and applications of synthetic human embryos, international collaboration and coordination are necessary. Harmonized and standardized laws and guidelines should be developed, along with increased transparency and accountability for practices and outcomes in this field. 3. Balancing risks. Striking a balance between the opportunities and risks of synthetic human embryos is vital. These embryos require respect and careful handling, as they have both benefits and costs for humanity. Prudent decisions and responsible oversight will be crucial in guiding this field's future. Conclusion. Synthetic human embryos represent a frontier of science teeming with promise and peril. They hold the potential to deepen our understanding of human development and advance healthcare, but they also raise ethical dilemmas and societal challenges. As we delve into the realm of synthetic human embryos, it is imperative to approach their creation, use, and regulation with caution, ensuring that they contribute to the greater good while respecting ethical boundaries. These developments implore us to reassess our perspectives on human life, redefining our views and values in the process. Thank you for watching. Subscribe to Explanium today and discover a whole new world of knowledge and understanding.